Hey guys, and welcome to our exam for review for 1406. So in this video, I'm going to use our study guide as a backbone, and I'm going to show you where to find all the answers and maybe even do some of the Mendelian problems that will pop up. So once again, as a disclaimer, this is not a comprehensive or like exclusive list that is going to be everything on the exam, but it's a good place to start. And then also don't forget about all the supplemental videos that are on Canvas, as well as all the supplemental handouts and everything. So definitely take a look at those. So let's go ahead and get started. So we're going to start with number 1. So it says make a chart of the phases of meiosis and what happens in each of them. So with that, I'm going to pull up our lecture 10 slides. So with lecture 10, it goes over all of meiosis. So a good chunk of meiosis should remind you of mitosis. So let's find some overview. So this is the overview of meiosis 1. So remember, meiosis goes 2 times instead of just once like mitosis. So in meiosis 1, we have prophase. We have prophase 1, metaphase, anaphase, and telophase 1, followed by cytokinesis. So in each one, it's a little different. So the big difference in terms of meiosis and mitosis is that in meiosis, in prophase 1, we have something that happens, it's called crossing over. So this is where the DNA will exchange whenever they touch. So whenever homologous chromosome pairs, so this is a chromosome and then here's another chromosome. So these are a homologous pairs. So whenever they line up, they touch, they will exchange genetic information. And that is how we get our genetic variability. It aids in that. So that happens in prophase 1, whereas in mitosis that does not happen. And then also what happens, the difference is that we have homologous chromosomes at the end of meiosis 1, whereas in mitosis we don't have that. So, but everything is the same in terms of metaphase, everything lines up in the middle. Anaphase, they get separated, and then telophase, you have 2 cells that are formed. Okay, so then now let's go into meiosis 2. So meiosis 2 is going to pick up right where we left off with 2 cells. So what's going to happen is that in prophase 2, the difference between prophase 1 and prophase 2 is that crossing over will not occur. Crossing over only occurs in prophase 1. And then in metaphase 2, they're going to line up along the metaphase plate just like normal. Same thing with anaphase 2, they're going to separate. And then in telophase 2, they're also going to separate and form new cells. And these cells are, they're haploid, they're not diploid. And then followed by cytokinesis, just like normal. So that was a very, very quick overview of meiosis, but definitely go and look at the in-between slides, such as these, just so you can get a better, deep understanding. Okie dokie. So with that, let's go and talk about what is crossing over and what's the result of crossing over. So we actually already kind of did that, but let me go back to this slide. So crossing over, like I said, it happens in prophase 1. This is where the homologous chromosomes, they pair up. So this is where one chromosome will find its homologous chromosomes, and then together, I'm sorry, not its homologous chromosome. Whenever they're together, then they form one homologous chromosome. And then whenever that happens, whenever the 2 different ones, so the non-sister chromatins, meaning that they are 2 different chromatins, they touch, and they touch at this chiasma or chiasmata. Whenever they touch, the chromatins, they become recombinant, meaning that they combine, they recombine, and they essentially swap. They swap genetic information, and some of the red will become on the blue, and then some of the blue will be on the red. Now, crossing over will occur in almost all chromosomes at least once or up to 3 times. That's pretty the excuse me. Excuse me. That's pretty much the norm in humans, but crossing over is random as to where it will take place. And then also how many times is going to be random. So there might be like one chromosome out of all of our 22 somatic chromosomes that may not cross over. So, which is perfectly fine. As long as we have crossing over that is occurring, we're good to go. But if crossing over does not occur, sorry guys, my throat's a little dry. As long as we have crossing over occurring, then we are going to increase our genetic variation, which is good for us. Now, some organisms it may not be as good, but for us it's definitely very much advantageous. Okie dokie. So what are the similarities and differences between mitosis and meiosis? So that is also found in our chapter, in chapter 10. So let me find that slide. So this is the big picture as to what makes meiosis different. So what makes meiosis different is that crossing over occurs. The alignment of homologous pairs are on the metaphase plate, whereas in mitosis, they're not homologous pairs, they're just chromosomes. They're just pairs of chromosomes. And then separation as well. So that's the big picture difference between meiosis and mitosis. But this is an even a little bit more in-depth version of it. So definitely that table in red is very, very important. So definitely look at that. That could definitely be a test question as to like filling in a table, since y'all know I'm very big on complete the table and everything. So definitely take a look at that. And let's go ahead and go to the next question because you can pause the video. So describe 3 ways that variation is increased because of sexual reproduction. So that is also found in chapter 10, I believe. Yes. So we have 3 things that contribute. So we have the first one is the independent assortment of chromosomes, followed by crossing over and random fertilization. So I'm not going to talk about crossing over again. I'm going to go to independent assortment and random fertilization. And then also remember that there is a video embedded in this lecture, so definitely take a look at that. But overview of independent assortment, this is where those homologous chromosomes in meiosis, they will randomly line up. So this happens in prophase where they get attached to their spindle fibers. So the order of which they get attached to their spindle fibers is also completely random. So, and then whenever they migrate to the middle, that is as well as random. So that's independent assortment, where they line up in metaphase, it's random. Okay, so like I said, I'm not going to talk about crossing over again, but random fertilization. So random fertilization is exactly what it sounds like. It is a complete chance as to which egg is going to get fertilized. So sometimes women, they will, whenever they undergo ovulation, meaning the egg is being released, that's roughly about 2 weeks before they have their menstrual cycle, they will release an egg. Sometimes they even release more than one egg, just depending on the woman, or sometimes they actually don't release an egg at all. But on average, let's just say a woman releases about 3 eggs every ovulation cycle. So it is complete chance as to which egg will get sperm in it first. So, and then also it's complete chance as to which sperm will actually penetrate the egg first. So that is where random fertilization is taking place. So we have 3 eggs. Let's say egg number 2 gets penetrated first by the 200 sperm that were currently on it, and only one of those sperm got through. So it's completely randomized. Now, if you get into like IVF and everything, that's where it's not so much random, because a lot of times a certain egg is selected, and then sometimes even a sperm is selected, depending on the situation in terms of the that patient case study. So if you get into IVF, that's not really true random fertilization. True random fertilization, if a male and female come together and they have consensual sex. That is along the lines of random fertilization, because the man cannot control which sperm gets there and the woman cannot control which egg is fertilized. So it's completely random. Okie dokie. So with that, we're going to go ahead and transition into Mendelian genetics now. So how did Mendel determine his law of genetics, and what are the 4 concepts of his model? So you can find that throughout lecture 11. So, oops, I'm gonna make this a little bit bigger. It's really small. Okay, so Mendel. So this is Gregory Mendel. He's known as the father of genetics because he's the one who actually came up with the basic principles of inheritance. Sorry guys, I don't know what is going on with my throat today. But what he did is that Gregory Mendel, he used pea plants in order to study genetics, and he traced the inheritance of various traits within the pea plants, and there were 7 that he looked at. These are the 7 that he looked at, and then whenever he was looking at that, he wanted to understand what made a certain pea plant look the way it did. And then by doing that, what he did is that he did true breeding between 2 parental plants that had different forms of the characteristics. So for example, the purple flower and the white flower. So true breeding, or pure breeding, whichever way, what it means is that the plant will always make more offspring like itself whenever it's self-fertilized over generations. And what I mean by that is this. So true breeding, so in this case we have a completely dominant parent, and then we have a completely recessive parent. So let me go ahead and write that. So I'm gonna just use A's. So completely dominant is capital, oops, that's really ugly, sorry guys. So completely dominant is capital, capital, and then completely recessive is little, little. So with that, if you were to cross these guys, so if you were to make your little Punnett square over here, so we have completely recessive and completely dominant. So this is pure breeding, whenever you have a homozygous dominant crossing with the homozygous recessive. So with that, we get big A, little a throughout the entire Punnett square. Sorry guys, my handwriting is really bad when I read on my computer. And then with that, this A, this capital A, will tell you what phenotype is going to be expressed. So with that, you make a completely purple flower in the next generation if you were to cross those 2 flowers. Now, if you were to cross so with this plant, so we saw that this plant has this genotype. So we have a big A, little a. So Gregory saw, we didn't do this. Oh no, I want to erase. Okay, I'm gonna just do it right below, guys. So if you were to cross the F1 generation, you will get 75% purple and 25% white. So that is how Gregory Mendel, he came up with his 4 concepts of genetics or of inheritance. So with that, I'm going to go ahead and state what they are. So the 4 concepts, so let me go to the first one. So the first one is the inheritance of a biological characteristic, or simply said, an allele. So these alleles, they're different versions of a gene. So meaning that for each character, an organism will inherit 2 pairs, one from mom, one from dad. And yeah, that's pretty much all I want to say about that. And then also those alleles, one will be dominant, one will be recessive for the most part. Or like what Gregory Mendel saw in whenever with his pea plants, he saw that once in a while you would get a completely recessive. You'll get two recessive alleles. So that is where you get the uncommon trait as compared to the dominant trait. So that's the first concept. And then the second concept is, gotta find it, guys, sorry, is the principle of segregation. So that is where two alleles, they are inheritable characteristics that will separate completely random. So this happens during the first phase of meiosis. Okay. And then I believe I have another slide on it. Nope, just kidding. Sorry guys, I don't have another slide on it. So that's the first one, the law of segregation. Okay. And then the other concept is law of independent assortment, which is right here. So the law of independent assortment, this is where you have each pair of alleles, they separate independently. So this is where they lined up in metaphase 1. So we've kind of already talked about that before. And then we have the law of dominance. So the law of dominance is where you have two alleles that are located in the same place on the chromosome, and then one will determine what does it look like. So as we saw previously with the purple white, so we saw that completely dominant and completely recessive. So in order to get a recessive phenotype, you have to have two little alleles in order for it to be expressed. But as we saw in the F1 generation, where we have a dominant and a recessive allele, the dominant allele will always be presented. You will always see that in the organism. But there is something called codominance, but we'll talk about that later when we get to that point in our study guide. So in terms of Mendelian genetics, we are just talking solely about his model, and then we'll talk about non-Mendelian genetics. Okay. So, so define dominant, recessive, homozygous, heterozygous phenotype and genotype. So dominant, it's exactly what it sounds like. It is dominant. It is going to be expressed. He is the dominant one. Recessive means that it's not always going to be expressed. So, such as this. So, dominant will determine what physical presentation the organism will have, and it's generally represented by a capital letter. And then recessive is the allele that is not noticeable unless it's paired with another recessive allele. And it's normally categorized or written with a lowercase letter. Okay, and then genotype and phenotype. So genotype is actually genetically what are you made of. So this is PP. And then whereas phenotype is actually physically how do you look. So if we have a capital P and a lowercase p, we are going to have a presentation of a purple flower. And then what were the other words? Homozygous and heterozygous, I believe. Yes, so homozygous means the same, heterozygous means one of each. So right here, the capital P, that's homozygous because they're the same. And then right here where we have capital P, lowercase p, that's heterozygous because we have one of each. Okay. Okay, so how did he determine the law of independent assortment? And the way that Mendel did this was through dihybrid crosses. So these dihybrid crosses, what they do is that they look at two different characteristics at the same time. So instead of just looking at one, you're looking at two. So this helped him figure out the independent assortment where each pair segregates independently during gamete formation or during meiosis. So he used dihybrid crosses, and then um, also just a little bit more information that the law of independent assortment, it states that each pair, like I said, segregates independently, and this law applies to genes on a chromosome that are not homologous. So meaning they're not found on the same chromosome, essentially, for the most part. Okay, so that was number 7. So let's talk about number 8. So number 8 is, what does it mean for a gene to exhibit plecotrophy? And then how does epistasis modify the Mendelian rules? And then what is a clue to a trait that may be due to multiple genes? So that can also be answered from chapter 11. Oh no, there it is. Okay. So let's go all the way down. So pleiotrophy, this means that one single gene affects multiple traits. So let me pull up a picture that kind of demonstrates this a little bit better. So pleiotrophy, so we have one gene and it can affect multiple things. So this one gene can code for feature 1, 2, and 3. Okay? So I hope that kind of gives a good definition or a good visual representation. So meaning that there is an allele that causes sickle cell anemia, and then a lot of times people who have sickle cell, they also have other conditions because that gene or that allele is affected. So a lot of times, sometimes sickle cell anemia, the allele, it can cause blindness depending on the person. It can cause liver failure, kidney problems, and a lot downstream effect. So that is pleiotrophy. How does epistasis modify Mendelian rules? So epistasis, so what epistasis is, first of all, is that a gene is located at one loci, and that it can alter the expression of the gene at the second loci of the same gene. So meaning that alleles of one gene may mask or conceal another allele. That's pretty much all I really want you to know about epistasis. I'm not going to ask you to do an epistasis problem on the test probably, but I'll definitely ask you about the concept of epistasis. Okay, so now it asks for, what is a clue that a trait may be due to multiple genes? So if a trait is due to multiple genes, it's also known as polygenetic inheritance. So this is where there are multiple genes that can control one thing. So an example of that is skin color. So skin color is a perfect example because there are multiple genes that control the pigment in a skin, which will dictate skin color, and it's specifically with the pigment melanin in the case of humans. So that is how that's kind of the little hints is whenever there are certain phenotypes, which is a continuum, meaning that there's different types of presentations, but it's all regarding the one single thing. Okie dokie. So now it asks for us to do some genetic problems. So I'm not going to do all of these. I'm only going to do the ones that I feel that are the hardest, which are, I believe, I'm gonna do B, C, and D. Okay, so for B, I'm gonna just enter all the way through so I have some writing space. Okay, so for B, it says, in radishes, a gene that controls color exhibits incomplete dominance. So pure breeding red radishes crossed with purebred white radishes will make purple radishes. What is the genotype and phenotype ratios when you cross a purple radish with a white radish? So before you even get started, you need to make a key as to what letters do you want to represent something. So in this case, I'm going to have C. So C with R and then CR. This is going to represent my red radishes. And then I'm going to have C with W's that are going to represent my white radishes. And then it said that whenever you cross these two, you'll get a purple radish. So meaning that if you have one from mom, which is like red, I guess, and then one from dad, which will be white, you will get purple radish. So then we set up our genotypes and phenotypes in order to start. So now we're going to cross a purple with a white. So purple, we said that our genotype is this, and then for white we said it's this. So now we're going to cross them. So we're going to get CR, CW, CR, CW, CW, CW. Okay, so let me actually make that R a little bit smaller. Okay, so then from that, we get, so genotypes and phenotypes. Sorry guys, that's really ugly. Eraser. And then phenotypes. So genotype wise, we have two of the same right here. So we have that 50% of the time. And then we have CWs 50% of the time. So 50%, 50%. So based on the phenotypes and genotypes that we set up before we started this problem, if we have a plant that has R and W, we're going to have a presentation of purple. And then if we have two W's, we're going to have a presentation of white. So 50% of the time, our population is going to be purple, then the other percentage, they're going to be white. Okay, so now let's do letter C. Okay, so Jimmy has AB blood and marries Tina with B type blood. The woman's mother has type O, and when Jimmy and Tina get married, they want to know what are the expected phenotypes and genotypes. So dad is AB. So if dad is AB, then his genotype is going, is going to be as follows. And then mom, she's type B, so she is going to be as follows. And the reason why she has a little I is because her mother is type O. And whenever you have someone who is type O, their genotype is automatically little I rather than capital I's. So with that, since that's her mom, that means automatically she got one of the little I's to her. Because technically, if you are also type B, you can be as follows. You can be big I as well, rather than big I, little I. But we know that this is mom's genotype because her mom is type O. Okay, so now we're going to cross mom and dad. So if we're going to cross mom and dad, so mom is this, dad is this. So you cross them. I personally put my A's first. You don't have to, but I highly recommend it. Okay, so based off of the Punnett square, you have a 25% chance of having the genotype being this one, and then you have a 25% chance of genotype being this. Sorry guys, I keep messing up on my letters. So, in this case, you have a 25% chance of being the following genotypes. Now, phenotypically wise, you have a 25% chance of being type AB, and then you have a 25% chance of being type A, and then you have a 50% chance of being type B, because we have a genotype right here and another genotype right there that codes for type B blood. Okay, now let's do one more, which is a sex-linked. Okay, so a recessive allele that causes a disorder called G6PD deficiency is located on the X chromosome. Beth has a history of the disorder in her family, but she does not have it. Beth's father has the deficiency. Jerry, her husband, does not. Their first child, Summer, does not have the deficiency, but they are expecting a second child. Rick, and they want to know the chances of which their second child will inherit the disorder. Okay, so with that, let's go ahead and set up our genotypes, and essentially just our genotypes for mom and dad. So mom, so she's automatically XX, and then dad is XY in this case. So dad is normal because it says that Jerry does not have the disorder. So he does not have a mutation on his X chromosome, but her mom does have a mutation on one of her X chromosomes. So I'm actually just going to put a little dot to represent the mutation on that X chromosome. And the reason why she has a mutation only on one of her X's is because her father does have the deficiency, but she does not. Okay, so now with that, let's go ahead and cross mom and dad. Okay, so we can see from this Punnett square that her daughters, if they have a daughter, then what will happen is that there's a 50% chance of their daughter being a carrier, but there is no chance of their daughter having the disease. Now with the son, if they were to have a son, then there is a 50% chance that the son will have the disorder. Okay, so the other two I'm not going to do because I feel that they are not as difficult and we have done some practice in class. But if you do have questions, definitely let me know. But let's go ahead and continue on. We only have 3 more problems and then we're done. So question number 10, it asks for what are sex chromosomes in humans and which gametes determine the sex chromosomes of an offspring, and why are X-linked disorders more common in males than they are in females. So let's answer the first question. So what are sex chromosomes? So sex chromosomes, they are the 23rd pair of our, I don't know why this froze. Sorry, guys. They are the 23rd pair of all of our chromosomes. So the first 22 are autosomic or they are somatic, whichever one. But our sex chromosome is the last pair. So if you are female, you are XX. If you are male, you are XY. So in the case of this karyotype, this is a male because it's an XY chromosome. Okay. Sorry guys, I gotta go back and look at the question. Okay, so like I said, we have 23 pairs, and the last pair, so the 23rd pair, is the one that actually determines our sex chromosomes, which are the differences between male and female. So X's are females, XY are males. Okay, and then it asks for sex-linked disorders. So sex-linked disorders are more common in men than female because the X chromosome contains more coding genes than the Y chromosome. And then also, a lot of times, as we saw in the previous problem that I just did, we see here that there's another X that can compensate for a mutated X. Whereas in males, they don't have another X to compensate for the mutated one. So that's another reason why it's more common in males than it is in females. Okay. That's the big picture as to why sex-linked disorders are more common in males than females. Okie dokie. So with that, let's talk about how is it possible for a gamete to have too many or too few chromosomes, and what genetic disorders will present with this. So, and then also what are some terms that go along with abnormal chromosome count. So first off, so how is it possible? So it is possible because there's something that can happen that's called non-disjunction, which is this guy. So non-disjunction, this happens whenever the chromosomes don't separate properly. So a disorder where chromosome numbers are caused by non-disjunction. So this is where a pair of homologous chromosomes or sister chromatins fail to separate in mitosis 1 or mitosis 2. And sometimes even in, I'm sorry, not mitosis, meiosis, but it can also occur in mitosis as well. So pretty much whenever homologous chromosomes or sister chromatins fail to separate, this is non-disjunction. So if it happens during meiosis, it will look something like this. So this is where it's in mitosis, whereas in meiosis, the non-disjunction happens in meiosis 1, right? I'm sorry, meiosis 2. So this is where too many chromosomes get pulled to the other side, and then that results in a wrong number of genetic material in the gametes. So there are a numerous amount of disorders that can present from this, and we're going to go to chapter 12 for that. I'm sorry, not chapter 12. Oh wait, yes, chapter 12. Sorry guys. Brain fart. So non-disjunction. So that happens randomly. It doesn't, there is some kind of mechanism, but it's completely randomized as to when non-disjunction will happen. And like I said, there are numerous amount of disorders that can go with this, and we'll talk about a few of them. But before we do that, let's talk about some vocab. So the vocabulary that goes along with this, sorry guys, is where you have like aneuploidy, which is where you have the incorrect number. So that's more or less just the overall term that we'll use to describe that there's incorrect number. Now, in order to be a little bit more detailed, what you'll do is you'll say monosomic, which means that there's only one particular pair, which is these guys. So these guys are monosomic, so meaning that they're lacking one. And then there is trisomic, which is where there's three particular copies, which is these guys. So these guys are trisomic. And these ones are normal. So whenever they have a pair, they're normal. And then ploidy just means that you have more than two sets. So you have triploidy, which is 3 sets of something. Tetraploidy means that you have 4 sets of something. So that's just the vocab. Now let's actually talk about the disorders. So some disorders that we talked about in class were the Turner syndrome. So Turner syndrome, this occurs in females. This is where they only have one X chromosome. Generally, these women, they are sterile. And then also, this is the only known viable monosomy in humans, meaning that the child, if this happens, they can still live. Okay, and then the other one that we talked about was Edwards syndrome. So this is a trisomy of chromosome number 18. So this happens in numerous ways. So this can happen whenever a gamete underwent non-disjunction, or it may even happen during the actual mitotic whenever the fetus is growing. It kind of just depends. But it is the second most common trisomy next to Down syndrome. Like I said, there are multiple types of Edwards syndrome, and each type will determine like the severity of it. But also with Edwards syndrome, depending on the severity, the baby can be very, very fragile. They're very small, and a lot of times they have a lot of health and physical defects to them, but they are a viable offspring. Now with where's Down syndrome? With Down syndrome, this is the trisomy of 21. So this one's also an aneuploidy in conditions of where you have too many or the wrong number of chromosomes. So this is where they have 3 copies of 21. Same thing with Down syndrome compared to Edwards syndrome. There are multiple severity levels of Down syndrome. So Down syndrome can happen whenever the gametes have the wrong number, or it can actually happen during the mitotic division whenever the fetus is actually growing, or it can happen, I believe, whenever there is a translocation of some genes. But not too concerned about y'all knowing that, but just kind of a fun fact. Also, Down syndrome, the frequency of Down syndrome will increase with the age of the mother. Now, the correlation hasn't exactly been explained, meaning like the mechanism as to why does that happen, but it might just, me personally, I think it's just because like cells get old. So as cells get old, they tend to make more mistakes because they're not young and chipper and everything. So as you get older, you tend to make more mistakes, and that's the same thing with your cells. As your cells get older, they make more mistakes. So it's completely natural. And then we have another one that we sort of talked about, which was Klinefelter's. So Klinefelter's, this is where you have an XXY. So this is these individuals, they are considered male. But with Klinefelter's, depending on like the severity of it, but for the most part, sometimes Klinefelter patients, they can have some physical and intellectual development, but for the most part, they're pretty normal. And also, they may be a little bit taller than normal. And also, these individuals, they are infertile, and that's actually normally when they figure out that they actually have Klinefelters. So for the most part, they do they do look normal, but then again, signs and symptoms will vary among patients. So each person is different. But they are infertile. That's actually normally when they find out because they're trying to have children, but then nothing's happening, and then they go to a doctor, and then they tell them like, "Oh, you actually have Klinefelters." Um, but yeah, for the most part, Klinefelters is not too severe. Now we do have something that is called trisomy X, which or also known as super female. So for the most part, these women they have no unusual physical features, although they may be a little bit slightly taller than average. But for the most part, they're completely normal. But just like with anything, there are some things that can go with it. Sometimes they develop completely normal, but there are some cases where there is increased risk of like learning disabilities. Um, Speech and language can be delayed. Also, they may even have some motor skills that can give them some like general muscle weakness while sitting or walking. Um, but like I said, generally, they don't have any issues. These they are fertile, but there are some cases where the women or the girls they will abnormally develop their ovaries and uterus. So with that, that can delay puberty. And then also with delay of puberty, that can also affect fertility down the line. But for the most part, completely normal and they're fine. And I believe that's all the conditions that I wanted to talk about. So let's go ahead and talk about number 12 real quick, and that will wrap up our video. So there are different types of chromosome structural alterations, and we're going to talk about each of them. And then also we're going to talk about how do each of these mutations occur. So just real quick, chapter 12. So alterations to chromosome structure. So as we know, chromosome structure is kind of like roughly like an X, but there are some things that can happen that can alter it. And there are four main things that we're going to talk about. There's deletion, duplication, inversion, and translocation. So let me go ahead and just pull up an image of that. So we have deletion. So deletion is exactly where it sounds like, where there is a part of the chromosome segment that is completely missing. So it's completely removed in everything. Sometimes, depending on if there's a large deletion that is missing, um, if it's a very big segment to the chromosome or a very vital part of the chromosome gets deleted, it can be lethal, meaning that it will no longer be a viable cell, or if that happens during mitotic division, whenever after fertilization has occurred, then the fetus will pass away. So that's for deletion. Now for duplication, this is exactly what it sounds like. This is where one or multiple segments are duplicated. So in the figure, it shows that B and C are duplicated. So instead of just being A, B, C, D, E, it becomes A, B, C, B, C, D, E. So duplication can also be very harmful depending on how much is duplicated and what is duplicated. And if too much is duplicated, then it can also be lethal because there's too much genetic information. And that can be detrimental to the gamete or as well to the fetus. Okay? And then we have inversion. So inversion is where you just flip something where certain segments of the chromosome are put in different orders. So as we see here, we have A, B, C, D, but then if inversion occurs, then we have A, D, C, B. So with inversion, normally it's okay because there is not a change in the number of genes. So that's generally it's okay. Phenotypes, they may be altered, but for the most part, the gamete as well as the fetus, if this happens during mitotic division, whenever the fetus is developing, it's okay. And then the last one we're going to talk about is translocation. So translocation is where two different segments, they will flip. So in this case, we have ABC, and then we have another pair, which is MNO. So this is where they are going to swap places on the chromosome. That can, depending on what is actually replaced, can can be harmful as well. But if it was the same amount, so like in this case, the first chromosome had 8 segments, and then if we ended with 8 segments, it might have been okay. But in this case, since there is one additional segment, the likelihood of it being not detrimental or harmful is minimized. So translocation, it can be harmful, but it kind of just depends as to what gets replaced, essentially. Okie dokie. So with that, I walked through the entire study guide, but once again, guys, this is not a exclusive list of what's on the test, but it's a good place to start. Don't forget to check out those supplemental videos and everything, those extra handouts. If you have questions, definitely contact. If not, I'll see y'all in class.